Welcome everyone. My name is Hemlata and I'm doing PhD at Department of Veterinary Pathology, University of Saskatchewan. Today I'm here to talk about the immunosuppression caused by infectious bursar disease virus and how it is impacting the necrotic enteritis in chickens which are raised without antimicrobials. So as we all are aware, necrotic enteritis primarily affect broiler chickens of 2 to 5 weeks of age. And once this disease established, it persists in the flock for 5 to 10 days. Clinically, a bird might show severe depression, ruffled feather, and sometimes brown foamy diarrhea. And there will be a sudden increase in mortality, which can go up to 50% in the flock. Necrotic enteritis led to severe economic losses to poultry industry in terms of poor weight gain, increased feed conversion ratio, increased mortality. And it was estimated that any cost around $6 billion every year to poultry industry. And these losses will keep on increasing as most of the countries, including Canada, is voluntarily withdrawing the use of in-feed prophylactic antibiotics. And chicken farmers of Canada have already withdrawn the use of category 1 and 2 antibiotics by the year 2018. And they are planning to withdraw category 3 as well in near future. So there is an imminent need to find the preventative strategies against necrotic enteritis. And here is one publication which emphasized the challenges of raising broiler chickens with restricted use of antibiotics in Canada, and which include top 3 concerns, includes coccidiosis, and necrotic enteritis is one of them. And we know necrotic enteritis is a multifactorial disease, as this bacteria requires amino acids because it cannot synthesize its own enzymes, and amino acid synthesis machinery is lacking. So when we fed chickens with the high protein diet, it's a source of multiplication for them. Another important factor is the coccidiosis, where it's direct damage to the intestines and help to multiply the C. perfringens in the intestines. And I would like to emphasize on the factor of stress, mainly because of the viral agents, immunosuppressive viral agents, which destroy the lymphoid tissues of the bird. And if we talk about immunosuppression here in Saskatchewan, this is one study from our lab where we have found that the incidences and economic impact of the IBD in the broiler production in Saskatchewan, Canada. And what we have found that 52% of the chicken barns and farms are infected with this virus. And Saskatchewan broiler chicken industry loses around 3.9 million kilogram of meat production per year due to this variant IBD strains. To study its impact under our experimental conditions, what we have done, we have divided birds into 2 different groups. First one, we have given the variant IBDV SKO9, in which we have 2 groups, set of 5 birds. One we have infected at day 1 of their age, another one we have infected at 15th day of their age. Along with that, we have the saline controls for each of the group. And then we have collected bursas and spleens from these birds for flow cytometry just to see the immune profile in those organs. So this is here the immune profile of the bursa fabricius. In the first column graph, we can see the B cell in the bursa. We can see in saline, it's about 90%, and it's coming down to 10 to 20% in the IBDV infected groups at both the time points. While if we go for the T cell profile, we can see it's increasing because in replacement of the B cells in the bursa fabricius. Similarly, for the spleen, we have seen the same picture. B cells are going down and T cells are coming in replacement of the B cells. So we have concluded that variant IBDV induced immunosuppression led to significant decrease in number of B cells in bursa fabricius and spleen, with significant increase in T cells response. So keeping this factor in the mind, we have developed an animal model for necrotic enteritis. So we consider 2 types of models here. One is subclinical necrotic enteritis, where we have just considered only the high protein diet change for the birds. And while for the clinical necrotic enteritis, we have considered both the factors, variant IBDV and a high protein diet, as predisposing factor for necrotic enteritis. So this is our objective, to develop a consistent necrotic enteritis model using these predisposing factors which are prevalent here in Saskatchewan. So we started with the screening of different necrotic enteritis C. perfringens isolates from the field. We have screened around 57 different isolates and have done conventional PCR for total 8 genes. And these all isolates were from different provinces of Canada, which I marked here on the map. And then we have found around 7 different combinations of the toxin genes in C. perfringens, among which we have chosen the one which has the most number of toxin genes and based on the literature. So further was our experimental design where we had 20 birds per group. First one, our control, no intervention. Second and third group, we have given C. perfringens feed with different level of protein, like one 20%, one is 28% in the feed, and challenge birds with the C. perfringens in the feed. While fourth and fifth group, we have same as second and third, only intervention is we have given IBDV along with the protein change. And sixth group, just to see the dose effect of C. perfringens, whether other groups we have given in the feed, and here we have given in the orally 1 ml morning and evening along with IBDV. So these are our different timelines here. At day 0, we place the bird. Day 17, we have given IBDV to these birds, and feed withdrawal before the protein changed to 28%, and then challenged these birds in feed and orally 2 times a day up to 5 days, and collected samples for histo just to see any lesions. So we were able to produce the disease in these birds, and grossly we can see the intestines were enlarged. When we cut open, there's a diffuse acute necrosis of the mucosa. And histologically as well, we can see the diffuse necrosis of the mucosa of intestines along with the C. perfringens bacterial colonies in the intestine. For mortality, what we have found, like the birds which we have given challenge with C. perfringens along with IBDV have around 10 to 15% mortality compared to our controls. And for gross and histo lesion, what we have found, despite of the different protein levels, the birds which we have given both predisposing factor have more necrotic enteritis lesion at both the time points, and which are around above 80% compared to the in-feed challenge alone with the different level of protein. So what we have concluded, like we developed a model of any in broiler chickens by exposing birds to C. perfringens following exposure to IBDV. And we have found incidences of necrotic enteritis are more when we are challenging along with the variant IBDV. And in this model, we have around 15% mortality and 80% of birds have histopathological any lesions. So this model we will use in our further studies for the prevention of necrotic enteritis. And another thing is control of variant IBDV will be very important in broiler chicken industry in order to control necrotic enteritis amid withdrawal of prophylactic antibiotics. With this, I would like to acknowledge our funding agencies, that is Poultry Science Cluster 3, which is a Canadian Agriculture Partnership, includes Agriculture and Agri-Food Canada, Canadian Poultry Research Council, and Chicken Farmers of Saskatchewan. Along with this, I would like to thank my supervisor, Dr. Susantha Gomez, for his guidance, and our research group for conducting those animal trials. With this, thank you all for your attention. Now I will be glad to answer any questions.
Thank you, Hemlata, for this very interesting presentation and the development of a new infectious model for necrotic enteritis. I have a question regarding your IBDV infection. If you can go back to your previous slide. And it has to do with your experimental model and the strain that you've used for your challenge. The IBDV, sorry. That you've used, sorry, not the Clostridium perfringens, because your strain of infection for the bacterial infection was very clear, but I'm curious about the infectious bursal disease strain that you've used.
Yeah, this is a good question. Like this isolate, before I came to this lab, they have like done a lot of work with this variant IBDV SK09, which is very prevalent in Saskatchewan. So it was like already standardized with the dose, like they have done the egg infective dose and stuff like that. So I have just used this one, like it's the most prevalent one right now circulating in the Saskatchewan barns as per the previous publications. So that's why we use this one.
And it's the very same one you used when you wanted to establish if it was better to go with a day 1 or a day 15 well, not to say what age was best, but when you wanted to look at the effect on your B and T cells, it was the same strain?
Yes, it's the same strain we used, and we have done like different time points earlier too. We have done one early age infection where we have challenged like 6 days around, which I'm not showing here. Another one we had around 17-day age. So we have seen like whether any kind of strain we are using, like despite of the age, it's causing the same depletion of the B cells in those ones.
Okay. And
Yeah.
again, going back to your local IBDV strain, how does it compare to the classification that is used for the various viral strains of bursal disease virus?
Yeah, it's this one is classified into variant one. Like we
Sorry?
have like this one is classified into the variant IBDV.
Oh, variant IBDV.
Yeah.
Okay, so and any closeness, I guess that you've done some sequencing, any closeness to other variants that could be present in Canada?
I'm not sure about that.
Okay. Oh,
Yeah.
that's okay. I'm just very interested by your model since our lab is working on necrotic enteritis. So it's very interesting to see how you've been able to reproduce well, not easily because it was a lot of work, but to reproduce your the disease in such a manner that it also represents the I guess what you see in the field. Because one of the challenges we see with the various necrotic enteritis infectious models that they're around is the fact that it's either it will cause too much lethality and mortality, which doesn't represent what you see in the field, or you see very low lesions and you need to rely on your histopathological changes in order to measure your changes in the efficacy of treatments and preventive means, so on and so forth. So kudos on putting together such a good good model. I'm wondering if there's any one from the audience who would like to ask questions to Hemlata. So I don't see any questions for the moment, but I was wondering what are the next steps and what's in the on the to-do list for the coming months with your model? How do you intend to be using it?
Yeah, like right now we have done so many. We are looking for some preventative strategies, like with some probiotics and vaccines. So we are on that path right now. So we have done like this one I presented is the preliminary work right now, but due to some publication things, I cannot reveal it, but we are like successful in some of the preventative strategies against necrotic enteritis right now.
Okay,
so
well,
yeah,
that's very interesting, and we look very much forward to your next publications and what is going to be coming out of your lab and Dr. Gomez's lab.